Okay, I'd like to introduce the second half of our presentation. This is Dr. Christopher Chitambar. He's a professor of medicine and biophysics, Division of Hematology and Oncology at the Medical College of Wisconsin. He's going to be talking to us about a new drug being used in a phase 1 study. Take it away. Wait, Chris, you're muted. Unmute yourself. Chris, you're muted. We can't hear you.
Can you hear me?
Now I can. I got you. Okay, start again.
Okay. Good evening, and thank you for joining us today, this evening. And as a follow-up we'll take you from one end to imaging and diagnostics to therapeutics. And I'm pleased to share with you a clinical trial that is an evolution that is actually about to be launched. So this is a trial of an agent that most people have not heard of. It's called Gallium Maltolate. And we will be studying this drug in patients with relapsed or refractory glioblastoma. And I am at the Medical College of Wisconsin. So Kathleen Shemenda, whose name you've heard several times in the previous presentation, is actually a collaborator of mine. And we'll go into this. So when one is dealing with a new drug, I think it's really important to put everything in perspective. So what I'm going to do is to give you a few minutes, several minutes, half the talk, will be devoted to talking about the preclinical work that went into this discovery. And then the latter part of the talk is actually going to go deal with the specifics of the trial itself and what we're looking for. So as this group, I'm sure, knows that glioblastoma, we're looking at high-grade glioma here. Really, the treatment has a long way to go despite modern-day therapy. Just the roadmap there to touch on is initial diagnosis, followed by surgery, current therapeutics here, followed by radiotherapy and chemotherapy. And despite this is the state of the art these days and may include temozolomide in most cases does. There may or may not be some other agents, but sadly, as you see in the right lower hand corner, data that it's not that old, shows that even in the best case scenario, median survival is measured in ranges in about the 14-month range. So I think that it's the latest data show that is where half the patients you get 14 months and that's where you're looking at having lost half the patients. So clearly, new drugs, new treatment modalities are needed for treatment of recurrent glioblastoma and glioblastoma in general. So coming to where we are and what led to this study is the fact that in the late 60s, early 70s, the Oak Ridge, Tennessee group there was looking at radioisotopes, and gallium-67 is a radioisotope, and when that was injected into animals, they were looking at ways to image tumors. And when gallium-67 was injected into animals, they found that lo and behold, it concentrated, isolated, or localized in tumors. And this was quite interesting, but that was the beginning of Gallium Scan, which has now been replaced by more modern day imaging technologies. But in fact, in many parts of the world, the Gallium Scan is still used to image tumors. And initially in the 70s, there was, when this imaging picked up steam, it was tested in a lot of tumors, and then it went from very quickly into the application in humans. Now, there was not an understanding in the early 70s as to why and how this isotope, this metal isotope actually concentrated. But subsequent studies actually done showed that gallium bound to a protein. Once it was injected into patients, it bound, the isotope bound avidly to transferrin. Now, transferrin is a protein in the circulation. It's normally present, and it's actually a protein that transports iron to around the body to cells. And as we'll get into in a few slides upcoming, but what was found that transferrin, if you had transferrin, it bound gallium, and you do have transferrin, and that led to targeting and rapid accumulation. And it was tested in many tumors, many patients, and what surfaced among the tumors was non-Hodgkin's lymphoma. So when one was looking at non-Hodgkin's lymphoma and staging these patients, the Gallium Scan was actually felt to be one of the important diagnostic tests for, so what you, for detection of residual or occult tumors. What you're looking at on the left hand of the slide, those dark areas are actually after the gallium, radioactive gallium is injected into a patient. And then there's a washout period. What's left are those dark areas, which are lymph nodes where gallium has accumulated. And what you're seeing is the outline, as you can identify, the head at the top and in the armpits, so the axilla. Those are enlarged lymph nodes, and lymph nodes down, lower down are lymph nodes in the groin. Well, this was the state of the art for many years for imaging. And what this cartoon here depicts is really how this is supposed to work. Now, transferrin, as I said, if you look at the lower half of the slide, you can see that transferrin actually physiologically, about 1/3 of this protein in the blood is occupied by iron. So iron, transferrin, there's no such thing as free floating iron in the blood. I mean, there is, but it's bound to something, otherwise it becomes very toxic. And that goes to tumor cells that have on their surfaces the transferrin receptor. And tumors such as lymphoma and other tumors, including glioblastoma, have very high densities of the transferrin receptor. And as a result, they need more iron. Well, gallium very much mimics iron in its chemical properties, except there's no need. Our bodies need iron, but they don't need gallium. But gallium will bind to transferrin, and it too will home in on transferrin receptor bearing cells. So gallium is a metal. It was discovered by a French chemist in 1875. And as I just mentioned, it does share certain properties that are similar to iron. Its ionic radius, it is 3+. It binds to transferrin in its lactoferrin and other iron binding proteins in the circulation, as well as iron proteins inside cells. It's unique. It's a unique metal, and if one looked at that, it really becomes, it's a solid compound, but you place it on their hand and it kind of semi-liquefies just at that temperature. Now, why is this so relevant to cancer? Well, a lot of research has gone into this, and early discoveries showed that cancer cells have a far greater need for iron than normal cells. So this is because there are a lot of processes, metabolism, cell division, and so forth that are amplified in cancer cells. And there's been a lot of work on this, some of which we've contributed to in my lab. And transferrin, actually, if you try to look at this in a cartoon form here, gallium is a mimic, is mimicking a mimetic, if you will, of sorts. iron by binding to transferrin, and you've got this Trojan horse approach here where the cancer cells thinks it's taking up iron, but in fact it binds transferrin, but transferrin, instead of containing iron, contains gallium. So the cell thinks it's taking up, through its transferrin receptor, iron for its utilization, but in fact it's taking up transferrin gallium. And as a result, you've tricked this cancer cell into taking up a metal that in fact will disrupt iron homeostasis in the cancer cell, simply put. But this in fact is step one. So early on, when a lot of my research was focused on looking at lymphoma, because I got into gallium research during my fellowship at the University of Colorado, and going back about 3 decades ago. And so there were early studies, though, when gallium scans were being developed for patient tumor imaging, was that it was shown that gallium actually was taken up by glioblastoma and brain tumors. Now, this was forgotten, as you can see the date on this in the Journal of Radiology, 1975. But here's the point being that in this slide is that gallium gets into the brain, gets across, because as we know, the blood-brain barrier is a big obstacle to current chemotherapy. The brain, it protects itself from foreign agents getting in, toxic. So it's a protective mechanism. But the blood-brain barrier may be partially disrupted for one reason or by brain tumors, but it may also be partially intact. So, but here's a good example of a lymphoma in a patient. And you can see that arrow pointing to the hot lymphoma spot in the brain. And after radiation therapy, which was a state of treatment for brain lymphoma in those days, and still it involved not chemotherapy, but there you go, you see that complete disappearance, making the point that gallium gets into the brain. So the first generation, when the discovery was made that radioactive gallium was taken up by brain tumors, there was a lot of excitement saying, well, what about non-radioactive gallium? And what about stable gallium salts? And so the first gallium, a number of them were tested at the NCI, National Cancer Institute, and the first gallium compound that was developed was gallium nitrate. And it's in fact a soluble gallium citrate nitrate complex. And it was designated an investigational drug, I believe, in 1979. So we're going back quite a ways with gallium nitrate. And there were a number of phase 2, phase 1 trials with gallium nitrate, and also in a variety of different tumors, but it was never tested in brain tumors, interestingly. But what surfaced from all those phase 2 trials, basically asking the question, is this active in cancers, was that gallium nitrate was found to have activity, anti-tumor activity in lymphoma and in bladder cancer. Those were among the few tumors that were tested. Some suggestion of activity in ovarian cancer. But the problem with gallium nitrate was that it has to be given intravenously. It's very cumbersome. There were different routes tried, and it was given as an intravenous bolus, meaning given over 15 to 20 minutes. And the pharmacokinetics of that were not the best, and you had to give it to patients continuously over 24 hours for at least 5 to 7 days. So you can imagine how difficult that is and how inconvenient for patients. And gallium, and everyone said, well, it'd be nice if we had a tablet, an oral formulation. So gallium nitrate is not bioavailable when it's given orally. It just isn't absorbed. So several years later, along came a compound, Gallium Maltolate. And Gallium Maltolate, I just refer to it as a second generation. That's not pharmacologically or technically what it's called, but I like to think of it as such. And in studies that have been done, some studies, just absorption studies, not full trials, but absorption studies have shown that in humans, and it is absorbed. And you can get comparable blood levels of gallium when it's given orally. There are, in fact, a number of different gallium compounds that are being developed, but this is the one that was closest to the clinic. So this is the basis. So there's a lot of iron in the brain, and initially we did some studies in my lab looking at glioblastoma stem cells, because I asked the question, and this is where actually I crossed Kathleen Tremaine and I, whose lab was actually down the hall from mine at the time, and it was serendipity. Kathleen was studying brain tumor models and in animals, and her postdoc was injecting drugs to try and looking at angiogenesis. And I one day said to her postdoc, I said, well, hey, you know, I'm looking at lymphoma, and I was trying to get a lymphoma brain tumor model going. So why don't you just take this vial of gallium maltolate, this in solution, and inject it in your animals? And she came back 2 months later and said, you want to see something interesting? It's shrinking, the tumors. And so this is what I'm going to lead up to, but this is our preclinical work. So in parallel with the animal work that I'm about to show you prior to the clinical trial, we looked at various glioblastoma cell lines. And as a result of that preclinical exciting discovery, and gallium maltolate, on the left, you're looking at cell lines, you're looking at the growth as the curve goes down. We're looking at D54, these are GBM cells, U87 human GBM cells, even a rat glioblastoma, and the HBME cells are the brain microvascular endothelial cells. So at the doses shown here, you can see that there's a straight line, there's straight lines that go across, and then there are curves that go straight. Well, in this experiment, it turns out that the endothelial cells, the microvascular endothelial cells, are not damaged at these concentrations, but we inhibit the cell growth in vitro. And you can see kind of the cell tissue culture in 2D systems here. And in collaboration with another collaborator, John Coe, who had developed glioblastoma stem cells taken from patients, he's a neurosurgeon. We did these grow as neurospheres, very closely mimicking the growth these are grown in suspension in vitro. We found that gallium maltolate blocked or diminished the neurosphere growth. And you can see that in the bottom where it says GAM, you can see those dark spheres, those are the spheres as opposed to the control. And though what you're looking at there are really, if you were to zoom in with microscopically and go down, that's low power, you would see that that's a cluster made up of multiple cells. So it's a tumor growing in suspension, but it's a three-dimensional growth. And you're looking at that change. And if you were to zoom in, it would look like a cluster of grapes actually in there, cells piled on top of each other. And you can appreciate the differences. And interestingly, you know, we've tried GSC, which is glioblastoma stem cell, different numbers just designated the arbitrary numbers we assign these cells, and we can see that there's inhibition right there. So very exciting in vitro. And so gallium, you can kind of look at what is it doing. And this is the result of much research that we've done looking at it. It acts, its mechanisms of action, it acts on the transferrin receptors, it disrupts iron utilization by tumors, it inhibits a key enzyme, ribonucleotide reductase, that depends on iron, and it also blocks mitochondrial energy production, induces cell death. Now, having done that, we then started thinking about a trial, and we're thinking the trial leading up to this now is there's precedent for the absorption of iron maltol. Iron maltol is in fact used in Europe to treat iron deficiency anemia. And so orally, gallium maltol or maltolate, actually, we're not quite sure how it gets across the gut. That's the enterocyte there. But it does, because in the trial, the human experiments that have been, human trials that have been done so far, you can give an individual oral gallium maltolate and then take samples in the blood, and it turns out that gallium is bound to transferrin, just like the radioisotope when injected, but oral gallium, 99% of it or 95% of it is found bound to transferrin. So it's mimicking iron transfer. That's a good thing. And then when you look at our model of how does this all work? Now, if you look at this cartoon here, we've tried to, so let me walk you through this. In the red, or the brown, that's what you're looking at the top part is the microvascular circulation of the brain, endothelial cells. Outside of the brain, the endothelial cells of the blood vessel do not have transferrin receptors, but the brain microvascular endothelial cells have transferrin receptors. And that's how they get their iron. Well, as I said, we're hijacking that system. And what's shown here, this is the immunohistochemistry, a slide from a biopsy of the brain, normal, just to show that in our hands and in others, the brown, dark brown staining actually represents the transferrin receptor staining here. Now, when you get inside the brain parenchyma, the cartoon here shows a glioblastoma. This again is a model. And glioblastomas also have high levels of transferrin receptor. In fact, if you look at immunohistochemistry staining, you'll see here the brown stain represents dark brown, and these are low power. You can see that this is all positive, meaning it expresses transferrin receptors. And so there's a pathway for transferrin bound iron to get into glioblastoma cells, but the red dots here, gallium transferrin is taken up at the same time. Whereas if you look at normal brain that surrounds the brain tumor, it doesn't, the glial cells, normal glial cells really don't have transferrin receptors. They get their iron differently. So you have here a great setup where you have in the glioblastoma cell, tumor has a target. The transferrin receptor is sitting in a sea of the brain, normal brain, and it's the island here. Now, when you get to this enzyme that is inhibited, ribonucleotide reductase, it's an enzyme that's expressed when in proliferating cells. Now, glioblastoma cells are growing. They're proliferating, the high-grade glioblastomas. And the staining here shows, again, that the brown stain, if you were to zoom in here where these red arrows are, actually stains for ribonucleotide reductase, specifically the M2 subunit that has iron center. So what it's telling us here is that GBM has a high expression of ribonucleotide reductase, which is a target for gallium. And we've shown in other studies that I'm not showing here that gallium inhibits ribonucleotide reductase, one of our early discoveries. Now, ribonucleotide reductase is not expressed in normal brain. And why should it be? Normal brain is not proliferating, but glioblastoma is. So we have a great scenario here where you have a target, you have an enzyme that's increased, and you have a compound, gallium, or a metal that will trick cells into taking it up and blocking the enzyme. Now, ferritin is where iron is stored, and in glioblastoma, you can see that ferritin is markedly increased, the brown stain here, compared to surrounding normal brain. So in short, what this model and the slide is depicting is that glioblastoma cells have a very high requirement for iron. And transferrin receptors, in fact, we have found in cells, and that see this green arrow, that glioblastoma cells actually produce transferrin, secrete it, and they secrete it as a mechanism into the parenchyma, and what they secrete it because they're trying to get more iron for their own growth. And gallium gets on that transferrin, and that's one more way. So it's a very highly proliferative tumor that requires iron, and is what we refer to as an autocrine mechanism. It's trying to produce a substance to help it get iron, and that substance happens to be transferrin. Very interesting. Okay, so we said this is fine as a model, but let's see if we can really show it works. And what better way to show it in a preclinical setting is to do an animal experiment. So these are live rats, and in collaboration with Kathleen Shemenda, who is studying these, we have been working very closely together. You can, at the Medical College of Wisconsin, the model we use is we inject, so it's an orthotopic, meaning the tumor cells, the xenograft is actually inoculated not under the skin or subcutaneously, but into the brain, which makes this a very unique model. So what we've done in this experiment here is that we have injected GBM cells, U87 cells, xenograft, and we inject it into the brain of a rat, immunocompromised rat, and we wait. It takes about 8 to 10 days, and then we do an MRI, brain MRI on this animal to confirm that a tumor is actually formed. So we don't start anything until we know there's a tumor. Then treatment, and in this experiment, we used intravenous gallium maltolate. This was one of the first studies we did. And we gave it, compared it with a control where we just used saline or salt water, placebo. And we treated this animal through a pump that was under, so this was continuously administered, indwelling pump that fed into the neck veins. And at day 18, so 10 days later, we took the animal, live animal, and did another MR to look to see what was happening to the brain tumor in that animal. And so the next slide actually shows this. And alluding to, now on the left panel here, you'll see that here's the brain, the glioblastoma growing in the rat brain, and that's the brain surrounding it. And then we also look at cerebral blood volume, basically some of the techniques alluded to in patients in the previous talk. And you can see that in the control animals, the tumor continues to grow unheeded. Now, in the gallium-treated animal, unfortunately, this does not project very well, but we started treatment and we found that the tumor grew much less slowly. So we use the word, it's retarded. The rate of growth is blocked or reduced. And so we statistically, we show that the percent change in tumors is from baseline is far greater in the untreated animal compared with the gallium treated animal. And this to us has led to a paper publication showing that gallium maltolate, when given intravenously in the animal, actually retarded the growth of GBM in an orthotopic model. Very exciting discovery, and I'm happy to say that no one else has done this study or shown this ever before. Now, we then said, well, that's nice, but what about oral gallium maltolate, oral? So we took gallium maltolate and we said, how do you give gallium maltolate to a rat? Well, clever research scientists in our group found that you can train a rat to take Nutella. Now you can buy Nutella in the grocery store. It's a concoction made of of oil, and it's like peanut butter of sorts. And what we do is we first train the rats to take Nutella, and they eat it and enjoy eating it. And then as next step, once the rat knows that here's something that tastes good, we embed it with gallium maltolate. 50 milligrams per kilo is the dose we've used. This is a fixed dose here based on prior studies. And you can see here that I'm showing you just currently a very striking example of what happened and in this animal, and then I'll show you actually what happens in survival. So this animal actually had this tumor, whoops, sorry, let me go back, growing, and it kept growing. And by day 50, we were treating the animal with oral gallium maltolate. And between a week here, the animal was looking like, was doing okay, even though this MR was showing this large enhancing tumor, which then we couldn't quite figure out what was going on. And so we said, okay, this is large enough. And then the animal starts showing some signs. And we said, we ended the experiment. And much to our surprise, when we did histopathology, we found this, where this tumor was supposed to be a tumor, we found that actually there was no tumor there, that this was just necrotic tissue in here, some vascular evidence for invasion around here, but really there was total resolution. What was appearing as tumor on imaging was no tumor, but and we've never seen this before. And we're seeing that this was the first ever, and we're writing this up. It's going to be published, I hope. But we've repeated such experiments in separate. So this, and of course, COVID hit at the same time when we had this. So you can see this was actually 2019, and then labs were shut down through 2020. But in the post-COVID, I shouldn't say post-COVID, but since things have begun to reopen, we have actually repeated experiments and found similar data. But it's very exciting for us to see that we actually, for the first time, I've never seen a tumor, established tumor this large, actually shrink down and disappear pathologically. But what about survival? The question we get, well, nice to shrink tumors, but do animals live longer? And in our results here, we actually found that not only do we shrink tumors, but in the cohort, of animals that are treated, we find that they live longer. And if you look at the days, you go from controls of 28 days to 51 days. Well, what does this mean? Well, in terms of humans, well, 1 month of survival in a rat is equal to 3 years of survival in a human. That's kind of where the model shows. So we're looking at significant prolongation if you were to extrapolate, and if this were to work in a human in a similar magnitude, we're looking at several years of life being prolonged just with this kind of approach. Now, this is single agent gallium maltolate, just one, and we're working on this, but this begs the question, shouldn't this be tested in humans? I mean, we've got a disease that's dismal. And so with that background, I'm happy to say that we are now in the process of launching. We're in the process of getting the IND from the FDA, and hope to, in the next few months, launch an oral gallium maltolate trial. It'll be a phase 1 trial. it'll be, and I'll end with a few, because we do have some background information on use of oral gallium maltolate. But the phase 1 trial that we are proposing, and this is actually registered in clinicaltrials.gov. So if you're interested, my colleague Jennifer Connolly is a neuro-oncologist, and she's listed as the PI on that. But you can search this at clinicaltrials.gov. The aims and objectives of this trial, being a phase 1 trial, is to really determine how well gallium maltolate is tolerated. So as will be shown in subsequent slides, we're going to start at a dose and move up on the dose. And we are going to try to define in the primary objective, how much of this is tolerated? What are the side effects of this oral gallium maltolate? And then, having achieved that, we will determine the safe and tolerability of oral gallium maltolate. And then we'll be able to use that as a recommended phase 2 dose. And then we're also going to be looking, because we'll be doing MR studies, sophisticated advanced MRI technology here, to see if in these patients, because who have recurrent glioblastoma, you know, the prognosis is not good. So we will know time enough if if we have impacted on a progression-free survival, if we've seen any anti-tumor activity at the maximally tolerated dose, and then we can also look and see if any this prolongation. So the primary endpoint is to define the toxicity, come up with that using standard, and the secondary is to use our imaging using well-established criteria. If there's toxicity, well, who's eligible for the trial? Well, obviously, we're looking at recurrent, so they must have a grade 4 glioblastoma, WHO classification, recurrent. All patients, regardless over the age of 18, are eligible. Consent, and they must be formed. Patients must have progressed after standard chemotherapy, and if they've had temozolomide, they must have completed that treatment 4 weeks prior to starting gallium maltolate. There's really no limit on what prior treatments. So if they've had other experimental treatments for their recurrence and now they are progressing, they're still eligible to go on trial. They must have measurable disease that we can actually define using, determine using the RANO criteria, which is an abbreviation for response assessment in neuro-oncology. They must have a reasonable clinical performance status, and they must have good kidney function, hepatic function, blood counts by the usual criteria, and must be postmenopausal. Almost, if they're premenopausal, they must agree to, in women, must agree to acceptable contraceptive prevention. In males, must use either be sterile or use barrier contraception since their previous drug dose. So it's very not restrictive in its eligibility criteria. There are no medications. They must have a certain lung function criteria and basically not have heart failure that's not being treated. There are no drugs as such that we know that would interact with gallium maltolate, but we do, because we know that there's a mechanism of action that involves iron. We do ask patients to not take iron supplements if they're taking multivitamins with iron or iron tablets, that they not take those while on gallium, nor should they be on any iron chelator therapy, because there's a possibility that gallium, being a metal like iron, could also be chelated if they're receiving an oral iron chelator or intravenous So the schema is this. We're starting patients, as I said, in a phase 1 study, it's a dose escalation. So we're starting with a daily dose of gallium maltolate at 500. And I'll explain why we picked 500, because there's previous evidence that 500 is well tolerated. And then we're going then to the next dose level, which will be 1,000 and next 1,500. And we're going through 3 dose levels. It's possible that if 1,500 is well tolerated, we may go higher. But the goal here is to define the recommended phase 2 dose, RP2D, for a phase 2 study. How many patients do we need? Well, we start at the lower dose and the first arm, and we do 3 per arm, 3 to 6 patients. If patients, DLT stands for dose limiting toxicity. So let's just say that we start at 500 and patients tolerate it well, and there's no real reason not to go up. Then we will expand and go up to the next dose level. But if we have 1 of 6 patients at a particular dose level that we're concerned is experiencing some toxicities, and we want to look at its harder, then we go to 6 patients. So it could go anywhere from 3 to 6 at each level. And if it's 1 of 6, only 1 of 6 seems to have toxicity problems, then we will go to the next level. So this is, but we do have de-escalation built in. So if, say at 1,500, someone's having problems, we'll go down to 1,250. So there's a de-escalation plan built into this. And the study calendar, this is a very busy calendar, but basically what it's showing you is that we do a pre-screening within the 30 days prior to enrolling patients. If they meet with all those tests, if they meet the criteria, then they start day 1. It's a 28-day cycle, but actually there's no interruption of gallium maltolate. They're going to get it every day at the dose for 2 months, so 28 times 2. And they're going to get a brain MR prior to starting, and then they'll get a brain MR at the end of the 2nd month. And if they're progressing on treatment, then obviously we will have assessed the toxicity. We'll have said, well, there's no signal. Remember, this is not a phase 2 trial. We're not going to make firm conclusions about does it work or does it not work. The goal here is to define, but we will be looking for any signals that might suggest, yeah, maybe there's something, but it's not a conclusion. And in a phase 1 trial, the phase 1 trial is really designed to determine what the dose might be for a larger trial, because statistically we need more patients to make conclusions on activity. We can just say signals, looks like we may see something. We're measuring all these things listed there, blood tests. Most of this is standard of care. You know, patients come in, whether it's this study, they get a brain MR, they on things that are out of standard of care is that we get an EKG and some lung function tests, and we measure gallium levels. But this is pretty much standard of care. How do we assess response? Well, we first look at, of course, organ toxicity. We're following very standard terminology to assess, because we will be reporting this as using the NCI common terminology and criteria for adverse effects, maximum tolerated doses, brain, as I said, MRI, and we'll be doing some additional studies that are more looking for markers, such as are there changes in the proteins of iron metabolism and in the blood with this. We're going to go back and look at the original biopsies to see did these tumors have high expression of transferrin receptors? Some may not. Or were there other things like ribonucleotide reductase expression? Was it really high in the original tumor, or was it not? Because that might give us some idea of if something looked good, if it turns out that patients that had very high ribonucleotide reductase, M2 expression, maybe those were the ones who might be more responsive, things like that. Again, exploratory. The study, like every study is going to be monitored closely by a data safety monitoring committee. All the regulatory compliance, ethics, and study management is pretty standard stuff that we do in phase 1 studies. Well, let me give you a little whiff of something that has stimulated this. There is some precedent, which I'm happy to say. Oftentimes we go into novel drugs and we say, oh, we don't know if this is going to do something or not, or is it even safe? Why do you start at this dose? So 20 years ago, nearly, this got, as I said, no one ever looked at brain tumors, but 20 years ago, there was a preliminary phase 1 with oral gallium maltolate done in patients. And medical college, one of my colleagues, Dave Wiesel, there was a study that was done by a company, but the company ran into financial problems and abandoned their gallium maltolate trial. But they did present this. So I can show this data because this is in the public domain. They did a study of gallium maltolate and they just used different cancers. They had 50 milligrams, 150 and 500 milligrams that they reported on. But they even said that they had one patient who tolerated 3,000 milligrams, but they don't have the data there. And these patients had prostate cancer, bladder cancer, all resistant to treatment, multiple myeloma, lymphoma. So you can see about 54 patients. And it looks like they gave these doses across, and they found that most of the patients, over 50% of the patients, had mild, what we call grade 1-2 toxicity. And the remainder had some, what they grade toxicity. When they say moderate, they're talking about grade 3 and 4. But what kind of side effects did they encounter? Well, in 3 patients, so it's really low. Out of 14, there were only 14 patients actually had grade 3 and 4. The side effects were seen in one of the 14, like constipation, some pain. Some patients had worsening of pain. They couldn't quite, so these are not bone pain now, back pain. It could be these are patients who had metastatic prostate cancer. So one patient had some complained of shortness of breath, respiratory distress, but there's no more information. And this patient, whether the patient had metastases or there's just no more information. That's why we're getting lung function tests on our patients. The thrombocytopenia, which means low platelet count, it turns out that of those patients, they actually had bone marrow involvement in prostate cancer. So it could be that it was the result of that, but we're monitoring patients, again, 3 out of 14 patients. So all in all, I think that from our perspective, we anticipate we have to do the phase 1 trial to be certain, but we don't anticipate really that we're going to run into severe side effects from this drug. It's very well tolerated from anecdotal experience and in just communications with the company. Initially, they said, oh, it was very well tolerated. And the trial was actually going to go with higher doses of gallium maltolate. But as I said, due to financial issues, the company had to discontinue it, and they don't have a gallium maltolate program, and they knew nothing about brain tumors. They went to 500 milligrams and didn't see much of side effects. Our study picks up where they left off, and it's brain tumors. So it's never been looked at in brain tumors. We have preclinical data that suggests that it's active in animals, and therefore we feel that this is a good study to go after a population where we have limited options. And I'm going to end on that note. I want to acknowledge my collaborators. Mona Al-Ghazawi, who's done a lot of the current animal work, Kathleen, who is supervising, Janine Worley, Hisham Al-Ajala, both of them in my laboratory. Jennifer Conley is going to be the collaborator. She's a co-principal investigator on the clinical trial. And I'm happy to take questions. Please feel free to email me at that address. Happy to answer stuff offline as well. So I'll stop there, Al.
Oh, thank you so much. This is very, very interesting. There's a few questions. First, did you give any thought to doing like a phase 0 type of study where you would give the drug, do a surgery and biopsy to make sure that the drug gets into the target tumor?
Yes, we would have to, we're actually assessing those studies, but, we're seeing, phase 0 in the tumor, it would require actually rebiopsy of the tumor of the patient.
Right. The next question was going to be, when people have recurrence, a lot of them get a surgery anyway. So where would the trial fit? You have to wait until after the surgery heals?
Yeah, we'd want them to recover from surgery. If they have a rebiopsy, that would be actually ideal in the sense that we've confirmed. And we can also measure the markers that I talked about in the last slides. We could look to see if they had markers, the transferrin receptor, ribonucleotide reductase, proliferative activity. Those would be actually very valuable in knowing, because as in most cases, if you have a marker, because I said not every patient may have a marker, but if we have tissue of the recurrence per se, then we can be sure that we have something that we can correlate for future studies downstream. Yeah, but it's not a requirement to go on the study.
Okay, I just want to remind people you could also ask questions about the FTB maps. We're going to mix the questions here now, but we don't have any right now yet.
Sure.
The next question is about the intellectual property rights of this drug. You said a drug company has it. And do they own it right now?
No, a drug company does not have it.
Is it public domain?
Exclusively, the person who has this is Larry Bernstein. And Larry Bernstein is...
So he acquired the rights to it. Okay.
He has it, and he is actually providing us with gallium maltolate. Yeah.
Okay. It's not available commercially now in any format, is it?
No. We have a company, GMP grade gallium maltolate. We've actually done our studies with that, and that is what is being evaluated for IND by the FDA.
Perfect.
And, yeah, in fact, they're reviewing it as we speak.
Okay. You mentioned the U87 model of rats. The question is, when you inject the brain tumor into the rat, what percentage of the rats actually get a brain tumor? Is it most of them or all of them?
Most of them. Most of them do. I couldn't give you a percent. I'd have to confirm, but most of them do seem to have a take. Okay,
there was another question. The presentation you made with the cartoon with the receptors was great. What about other ways to target those receptors? Are there any other ways? Like you're talking about using the gallium maltolate, like this one right here. Could you have like a vaccine against these receptors?
Yeah. So
like a CAR T cell?
So the transferrin receptor strategy, using transferrin to try to get to target and use that pathway, has been tried by other people. Other investigators. Some people have tried a vaccine, some people have tried to deliver a virus, some have tried to link other chemo drugs to this. So to targeting the transferrin receptor. So what is different about ours, so they've tried that, they've tried to put, but you know what the difference there is that they've actually made a transferrin drug conjugate and injected it. And so far, there's been toxicity issues. Now, because, you know, whether they've linked a toxin to it, there's an external transferrin being injected. But so yes, the pathway, and if one searches trying to exploit the transferrin receptor, that's true. That has been tried that has been tried with other therapies, toxins, antibodies, and so forth, coupled to it. So yes, that has been tried. What's different about our pathway is that it's more a natural way, because what it's doing is we're not injecting transferrin. We're actually, gallium taken in orally is going to physiologically, or, you know, well, it's not physiologic because there's no gallium in physiology, but it will bind from the gut to transferrin that's already endogenous, you know.
Okay, outside of the brain,
yeah.
What organs have a high level of the transferrin receptor?
The liver and red blood cells.
Okay.
I should say not red blood cells, excuse me. Red blood erythroid precursors in the bone marrow. So interestingly, that's where, because iron, what's iron used for? Developing hemoglobin. So that's where red cell precursors, but not circulating red blood cells.
So if you found that this interferes with blood counts, you might need a way to deliver this locally.
So but if it interferes now with the gallium maltolate, the gallium nitrate study, some patients developed anemia. Interestingly enough, even though we're checking for white blood cell counts, gallium nitrate, when that was used, did not lower the white blood cell count, but it did make some patients anemic. And you know, but anemia is something that we can treat with red cell transfusions, you know.
Oh, sure.
So that's something different, you know. It's very much.
How long did you plan on using this? It's daily for how long?
Well, the trial, the study itself is 8 weeks.
8 weeks.
So 8 weeks, but now, you know, and before the second MR. Well, let's envision that, but if a patient at 8 weeks has stable disease and is tolerating the treatment, that patient has the option of continuing on.
Perfect. That's what we're going to ask next.
Up to 6 months. Yeah, continuing. I mean, there's really no endpoint. In someone who's responding, we will continue treating, or is stable, continue treatment, because that's the other side of it.
Okay, I'm just checking Facebook questions. Okay, the gallium is not related to gadolinium at all. It's completely different.
No,
right?
no,
Right, okay.
Absolutely not. No,
Okay.
Very different.
That's what I thought. That's all the questions that we have, and we have no more for the FTB maps. I'm sorry. This was very enlightening, and we'll be looking at this trial very closely, keeping an eye on it. It sounds very, very interesting, and if it works, it looks like you could get this going relatively quickly, because it's an oral drug, relatively, hopefully safe.
Yeah.
I wish you luck.
Thank you very much, Al.
I just want to tell people about our upcoming webinars. Next week, we have A New Hope in Treating Metastatic Brain Tumors. And that's from the Brain Metastasis Clinic at MD Anderson, which should be very good. And then the last one is going to be a special one about our organization. We're going to talk about our special projects like our patient navigation program, ClinWiki, which is a way to make it easier to find clinical trials. One way is we're looking at making videos about each clinical trial. This would be a perfect video for your clinical trial. So on our listing on our webpage for your clinical trial, we can link to this video so people could see why you want to do a clinical trial. And then the biggest thing that we've ever worked on is called the Promising Pathway Act. It's a new law designed to take drugs like what you're talking about and get them to patients much quicker. Basically, it would do away with the need for a phase 3 trial, and you would get a conditional approval after phase 2. You would cut off like 95% of the cost of developing a drug. But in return, every patient who uses a drug while it's conditionally approved has to be followed in a registry as if they're in a clinical trial. So it's like taking the phase 3 trial and putting it after approval. And insurance companies will be sort of required or encouraged to pay for the drugs. It's a big topic. The bill was just introduced 2 days ago, so we'll be talking a lot about that soon.
Yeah, I'd like to learn a lot about that one. Thank you.
Oh yeah, that'll help all the small people, I shouldn't say small, but all the researchers like you who have great, brilliant ideas. I see this all the time. They have great ideas, it looks great in the rat, And then they hit the problem of you now have to raise $50 million to run your clinical trials, and you can't do it.
You're
Now, if this bill passes, the brain tumor organizations could actually afford to take a drug like what you're talking about and put it through all the clinical trials to get the FDA approval without having to go form a drug company, without having to get all this money. And we'll get 5 or 10 new drugs a year and be able to see all the interaction between the drugs, figure out the best cocktails. For example, if your drug cuts down 75% of the tumor, you're still dying. But if you have 2 or 3 treatments that each cut out 75% of the drug, you might have a cure.
Well, I should just mention that we've actually recently published on this, that we've actually shown synergy, very strong synergy in, at least in vitro, we haven't done it in animals yet, between this drug, a gallium maltolate, and very frequently used drug, metformin.
Oh, wow. Okay. So that's,
I mean, it's truly remarkable that there's opportunities. This could be one that you could build on.
Well, not only that, but theoretically, your drug plus metformin should sort of rob the tumor of its energy and weaken it so that it makes it more susceptible to all the other treatments. So, and we have a team of experts that's part of our patient navigation program that looks at all these things and tries to come up with the best combinations. And we try inside a virtual trial using our registry. And there's no way to do something like that unless the drugs are approved. So that's why we need this conditional approval process. So we'll learn all about that 2 weeks from tonight. I'll see everybody there, and thank both of you, Chris and Mark. Thank you so much.
Thank you. Good night.
Good night.
Thank you. Thank you for the invitation.
Sure.